Okay, sound, if you recall, in the inner ear, you have your cochlea, this structure. The cochlea, and it's in this structure where you find your receptor for sound, or we call your organ of Corti. All right, so let's go into this structure. If we were to say unwind it this way, we would find 3 chambers in there. Okay, here will be the oval window. Here's a round window, oval window, round window. This top chamber is what we call your scala vestibuli. The middle one is what we call your scala media. The bottom one is what we call your scala tympani. So you have 3 chambers there in the cochlea. And in the scala media, also called the cochlear duct, this is where you have your organ of Corti. So on the base of it, on this this is called your basilar membrane of the organ of Corti. On there sits a structure. We have cells called hair cells. And these hair cells sit on the basilar membrane, and they have little things called stereocilia that stick up to anchor those hair cells, anchor them to another membrane here called your tectorial membrane. Okay. And these hair cells are linked to neurons. So when those hair cells are activated, they eventually trigger action potentials in these neurons, which make up your cranial nerve number 8, your vestibulocochlear nerve. All right, so let's dig into some more here. So again, so this part here is called the organ of Corti. So if we look at that part in some more detail, and here you have your basilar membrane, and on that membrane sits hair cell. This sits in 2 groups. There's a group of 3. There's a gap, and there's a single cell group, okay? And they run this way along the membrane, 3 and 1 this way. So the outer 3 are called the outer hair cells, these 3, and this single one is called the inner hair cell. So, and both sets of cells anchor themselves into your tectorial membrane, okay? For the outer hair cells, they all have equal length stereocilia. I'm calling them stereocilia. These extensions upwards. Stereocilia. Whereas the inner hair cells, the stereocilia there are not the same length. They're a graded length from short to long, tall, like that. And they're linked by a membrane called the tip link. So let's dig in some more on the inner hair cells. So the inner hair cells sit, again, on the basilar membrane, and they extend upward. Extend up. Put it right here. Inner hair cells here. Basilar membrane is there. These hair cells, like that, extend up, like so, with graded stereocilia, like so, and here the tip link. So the tallest stereocilia, this one, is called your kinocilium. This is the tallest one, and the rest are just called regular stereocilia. Again, this is your tip link. Now, if you really dig down on the tip link part, you will see that the tip link is actually a protein that can control gaits. So here, if you look at it in a bigger view, like so, you can see here, you are your kinocilium, like that, okay. Here's the tip link. It's actually attached. So at the tip, at these tips, you have ion channels like this. These are channels, which can be opened and closed by your tip link. So these ion channels will let in ions, of course, let in potassium and calcium ions. So these are what we call mechanical gated channels, because when the tip link pulls on it, that's when they open or close. So keep that in mind. And when these ions come in, they're able to depolarize the cell, which then leads to these cells releasing neurotransmitters, which then will trigger action potentials in your cranial nerve number 8. All right, let's dig in some more. So now we want to show you how it works. So let me erase this again and start from a different starting point. So here now, let's say at rest, like this. At rest, your cell is there. Okay. Now, of course, your cilia, that is there. Here's the tip link. So at rest, some of these channels are open anyway. So there is some casual leaking in of potassium and sodium. Of potassium and calcium. And that slow leaking of these ions will depolarize the cell to create a basal firing of action potentials in the neuron. So this is at rest. Now when you vibrate, vibrate up and down. So as you vibrate, sometimes these cells will go up. When they go up like this, so here now is basal membrane is there. The cell go up like that. The membrane, it's still here. This is your textural membrane. So now the cells will do this. They bend this way. Bend, because they're being pulled. Because when the tall one bends, the tip link pulls the other cells open, like so. And that causes them to open up wider, so then more ions jump into the cell. So more Ks, more calcium ions pop into the cell when it goes up, and that will cause you to have more action potentials. Okay, so this is rest. Here you have increase in action potentials, and then when it vibrates the other way, downward this way, so now the membrane, the basal membrane comes down. This one is still there. So in essence, I'll make myself too big here. These stereocilia now are stretched. And when they're stretched, they close. When they close, you have less ions coming in, and so in essence what happens here is that in this case, when they go down, they fire less. So here you have a rest, basal level of action potentials, goes up, fires a lot more, goes down less. AP fires in that neuron. And that's how the brain knows what's coming in, by how often it goes up and down. That kind of codes for the frequency, as a matter of fact. Okay? So it knows that there's activity there. So it's like an on-off system. It's not on, it's on-off. Up. Fast or slow, depending on how fast the membrane is vibrating. That's how you create your action potentials in your auditory nerve. Okay, so let me see what else I got before I stop there. Okay, so we'll pause there.